Hey guys, it's Miss Johnson. I just wanted to go over the review sheet with you that I put on Canvas today. So it's your assignment today is to work on this sheet and get ready for your test that's going to be tomorrow. And so I just want to go over some of the important things with you and make sure that if you need the help, you've got it. If you feel like you know the answers to these questions and don't need my help, and by all means, you don't need to listen to this video, but this is just to make sure that any questions get answered along the way. All right, so as you're working today, if you have questions, go ahead and email me, and I'll be happy to throw you back an answer as quickly as I can, and we can work through that and anything you're struggling on. So starting with our study guide today, number 1, it says, what are the ways that variation can happen in a population? And so some of the things that I think it was from the video the other day, there were guided notes that you had with the video and some of the things that cause variation. So number 1 is sexual reproduction. Okay, so know that just by having two different parents that make an offspring that's different from them, that's going to add variation to a population. So aside from, all the other things that we know cause variation, that in and of itself takes two things that are different, makes a new thing that's different from both of them, and that leads to variation. Part of that is also the process of meiosis, which is the next thing. So that process alone leads to variation because we talked about when homologous chromosomes pair up, they cross over, and that leads to variation. And then also when they line up in the middle, there's no organization on which one goes where in terms of the one from mom and one from dad. And so that independent assortment of chromosomes during meiosis also leads to variation. So there's two important things that are related to sexual reproduction, but two technically different things. And then the third thing would be mutation. And so we spent a lot of time in our last unit looking at mutation when you have one single base that changes in DNA, or when a base gets added or deleted in DNA, that leads to mutation. And so that's going to lead to variation. And not all mutations, remember, are bad. So some of them are good, and so that's what leads to all the different traits that we have within a population. So mutations are neither good nor bad. It's if they're fit for the environment. And that's actually what we'll be moving on to in our next big unit on evolution and natural selection. All right, so question number 2, it says, could things like change in water temperature cause a mutation in an egg cell of fish in a stream? And then what might be the result of such a mutation? So yes, environmental factors can cause mutation. I can't spell environmental. Okay, so we know environment can cause mutations. So things like UV radiation, we know that causes skin cancer, and that's a really good example of something in the environment that can cause changes to our DNA. So UV radiation, but even change in water temperature. If you've got an egg that's laid in a stream and the water temperature goes too high, that could cause a mutation. And so the next part of the question, what might be the result of such a mutation? Well, a whole bunch of different things. It could be a whole lot of different things. Maybe changes in fin shape or fin length or fish size or coloration. So all different kinds of things. Mutations are not predictable as to what might be mutated and what they might cause. All right. Number 3, it says humans have 46 chromosomes. How many somatic chromosomes are there? Remember, when we talked about karyotypes, we talked about the difference between like the somatic or body chromosomes versus the sex chromosomes. So if there's 23 pairs of chromosomes, gives us 46. Remember, 22 pairs are somatic. I cannot spell. Are somatic. Okay, so remember, 22 pairs would be 44 of them. So 44 chromosomes, individual chromosomes, would be the somatic chromosomes. Remember, you get one from mom, one from dad. The second part of the question, how many sex chromosomes are there? Well, there's 2. And so there's 2, I should redo this. So there'll be one pair are sex chromosomes, and then, so that's 2 total chromosomes. And so how do we identify the sex chromosomes? Remember that male is X and a Y chromosome, and and female are 2 X chromosomes. So if you're male, you're XX. I'm sorry, I said that wrong. If you're male, you're XY. If you're female, you're XX. So 44 of them, or 22 pairs, are what we call somatic chromosomes. They control most of our traits. And then that last pair, or 2 individual chromosomes, whether they're XX or XY, are the sex chromosomes. And so 44 plus 2 is 46, and that gives us our 46 chromosomes. And that number is important. That 46 chromosomes, that's how many we should have. Anything above or below that would be incorrect and cause potential issues. All right, question number 4. What would cause the number of chromosomes in a human to increase or decrease? And so remember, we showed during meiosis, sometimes the chromosomes don't separate right. And if you remember that term, it's non-disjunction. Okay, so I'm going to draw a quick picture here. So remember, when homologous chromosomes are paired up in the middle, when they go to split, so they should go, I kind of drew that funny. So when they go to split, sometimes a pair just doesn't separate the way they should. So this one over here should have separated and went over here, but it didn't. So it didn't just disjoin. So non-disjunction is when chromosomes don't separate right. So now this cell that would form after the process finishes would have a whole extra set of chromosomes. And so that, remember, would not be a good thing. All right, question number 5. Draw the steps of meiosis. And then the second part, which stages lead to genetic variation, and explain. So let's just go ahead and quickly, and I won't ask you to draw the steps of meiosis on the test, but if I give you a picture of a step in meiosis, you should be able to identify which stage it is. So thinking about our steps of meiosis, so I'm going to quickly do this. So we've got, remember, the homologous chromosomes kind of pair up and become visible, and that is prophase 1. And then from there, they move to the middle. So we got homologous pairs of chromosomes lined up in the middle, and so that's metaphase 1. So remember PMAT, prophase, metaphase, and then after metaphase comes, do we know? Too bad you guys aren't here to answer back, but I think you're saying it in your head. Hopefully it's anaphase 1. So we got PMA, and then the last part would be T for telophase 1. So telophase 1, we essentially have 2 new cells, and where we started out with 6 chromosomes in the first cell, now we have 3 chromosomes in each cell. I didn't give myself enough room here, so I'm going to kind of weave my way around. Okay, so remember, in meiosis, just remind us, in meiosis, remember, we have 2 steps. So we go through PMAT once, and then we go through PMAT a second time. So prophase, metaphase, anaphase, telophase 1, and then we start back in with prophase 2. And so in prophase 2, again, not much different happens, but we've got those same 2 cells with the chromosomes condensed so we can see them. And then after prophase comes, didn't draw those very well, metaphase 2. Okay, so we got prophase, metaphase. I know you're thinking, what comes next? And I know you already know that it's anaphase 2. Okay, and here, so we have individual chromosomes lined up in the middle, which is different than metaphase 1, where we have homologous chromosomes. So remember, these up here, when they're paired chromosomes like this, these are called homologous chromosomes. Okay, so we got homologous chromosomes that are paired up here in metaphase 1, but individual chromosomes, half the number of individual chromosomes in metaphase 2. And so in anaphase, then, it's not chromosomes that separate, but it's chromatids or sister chromatids. Remember, this is the duplicated DNA. So before each cell got to this point, they made a copy of that DNA. So each chromosome had two sides. And so we've got anaphase 2, and then our last stage is now, so we've gone through PMAT, PMA, and T is for telophase. And so at the end, we end up with 4 cells, and all 4 cells have half of what they started out with. So we started out with 1 cell that had 6 chromosomes, and here we ended up at the end of telophase with 4 cells that each have only 3 chromosomes, half the number. And so the second part of the question says, which stages lead to genetic variation? And so remember in prophase 1, that's where crossing over can happen. Okay, and crossing over leads to a huge amount of genetic variation because all those little pieces switch places. And then also in metaphase 1, remember all the homologous chromosomes line up, but there's nothing that dictates the way they line up, whether moms are on the left or dads are on the right. There's nothing that says that. And if you remember and you look in your notebooks, it's called independent assortment. Okay, and I happen to have my diagram that we did together that we put in our composition book, so hopefully you have this in yours, and if not, you know, you might want to take a minute and draw what you see there. But remember, we talked about in prophase 1, those chromosomes cross over and little pieces of the DNA switch place. Okay, so that's in prophase to metaphase 1. They switch places. And then we also talked about, independent assortment. So independent assortment, when the chromosomes line up in the middle, there's nothing that says that all of the blue have to be on one side and the pink on the other. They can flip-flop. And remember, you have 23 pairs of them, so they can flip-flop 23 to the 23rd different times. And so this is called independent assortment, and it makes all the gametes different. So when you take this switching places and this variation in the way they line up, that adds to a huge amount of genetic variation. So I know I'm backtracking a little bit, but if we go back to question number 1, thinking question number 1, that idea of meiosis leads to a lot of variation because of these two things, crossing over and independent assortment. All right, moving on. Question number 6. I think I already answered that, and you probably already know the answer. So it says, what special event occurs during prophase 1 of meiosis leads to variation of the gametes that are produced? And I already said it, but this right here, that is our answer to number 6, crossing over. So for number 6, that's the big thing that happens. So when those homologous chromosomes come together, crossing over can happen, and that leads to a huge amount of variation. Okay, and I'm not going to take the time to draw what crossing over looks like for number 7, but it wouldn't be a bad idea for you. So this is crossing over where the chromatids kind of get tangled up and parts of the chromosomes switch places. And remember, we tried to do this with Play-Doh. Tried. Okay. All right, so that's question number 7. Then what do they look like? All right, question number 8, what occurs during metaphase 1 to lead to variation in the gametes produced? And so this is the answer for number 8, independent assortment. So for number 8, this idea that we just talked about, independent assortment, that's what leads to a lot of variation. So again, I'm not going to draw it. You can take the time to draw it if you want. But remember that for all of our 23 chromosomes, there's nothing that says that all of the dads have to be here and moms have to be here. They flip-flop. So take this times 23 times all the chromosomes that would line up in the middle during your metaphase, or when metaphase happens in your body. Take all those 23 chromosomes, and there's nothing that says which one has to be right and left. And so that independent assortment, that's the second thing that leads to a huge amount of variation. So number 9 says draw what it looks like. Well, I'm not going to take the time to do that because I had the diagram there for you. All right, moving on. Let's look at question number, what are we on? Number 10. How do things, let me write that down. So number 10, how do things like sickle cell anemia and cystic fibrosis occur and get passed from parent to offspring? Okay, so this kind of ties in what we learned in our last chapter with what we're learning, or not chapter, but our last learning target and this learning target. So sickle cell anemia and cystic fibrosis, can't talk today, all occur because of mutation. Okay, and so remember back when we looked at sickle cell a little bit, it's one specific letter that changes in the DNA that changes the protein, changes like a glutamine to a valine. So one single amino acid changes, and that changes the whole shape of the protein. And a similar thing with cystic fibrosis. So one little change in the DNA, I'm going to put change in DNA. So one little change in DNA can cause something like sickle cell or cystic fibrosis to happen. And so the question is, how does it get passed from parent to offspring? Well, if something happens and it's part of your DNA, then that DNA has the potential to be passed on to your offspring if it's part of your DNA that makes up your egg and sperm cells. And so if this change, if this mutation occurs from the very beginning, then it's going to end up in all of their cells. All right, number 11. If a mutation occurs in a brain or skin cell, can it be passed to the offspring? And so this kind of leads from that question that I just asked. And so the answer is no, an emphatic no. So things that happen to you during your lifetime, in your hair, your skin, your pancreas, your liver, all those parts of your body, those mutations don't get passed on to your offspring. Okay? So think about it. If something happens in your skin, is there any way for the skin cells to be carried on to your offspring? And the answer is no. So remember, so body cells are somatic. And that's an important word. So when we talk about somatic cells, we're talking about body cells. And so any mutation that happens in a somatic cell, it's not going to be passed on. Now, it has the potential to cause cancer or cell death. Okay, so a mutation in your body cell could cause cancer, it could cause that cell to die, but it would never be passed on to your offspring. All right, number 12 then. If a mutation occurs in an egg or sperm cell, can it be passed to the offspring? And explain. And the answer is yes. So anything that happens in an egg or sperm cell, and so we call those sex cells or gamete. Oops, I cannot spell M-E-T-E. Okay, so anything that happens in a sex cell or a gamete can definitely be passed on to the offspring, because those are the cells that come together to form the zygote. So anything that's going on in this egg or sperm cell has every potential to be passed on to the new offspring. All right, number 13. If a new trait appears in a population, such as a pink flower when the rest are yellow, or a white mouse when the rest are brown, what is the most likely cause of such event? So you've got all yellow flowers and all of a sudden there's a pink one, or all brown mice and all of a sudden there's a white one. The thing that's going to cause that thing to change, that one individual to change, is mutation. So if you see a new trait appear in a population, that's almost always due to some sort of mutation. And so mutations happen, and so if it's a mutation that happened in an egg or a sperm cell in a previous line, then that has the potential to cause a different organism, or an organism with a new trait to appear. And then number 14, Mutations can happen in any type of cell. What is different about the effects of mutations that occur in somatic cells versus gametic cells or gamete cells? And so basically we just talked about it. So that's just a review for from what we talked about in these previous questions. And so if a mutation happens in a somatic cell, it's a body cell. So it has a potential to lead to cancer or cause cell death or cause the organism to not function well, depending on where the issue happened or the mutation happened. If a mutation happens in a sex cell, an egg or a sperm cell, it has the potential to be passed on to the next generation. It could be a good thing or a bad thing. So remember, mutations are neither good or bad. So that's important to know. So mutations are not necessarily good or bad. So when a mutation happens, if it's a good thing, then it'll stay around and become part of the population. But if it's a bad thing, then it causes something like cystic fibrosis, and maybe that's not a good thing in the population. And so that's basically all of this together is your answer for number 14 here. So just kind of wrap that up. And I will tell you on your test tomorrow, this number 14, this is what you're going to need to write about. So understanding that mutations happen, and depending on where they happen, whether they happen in your somatic cells or your sex cells, they can have potentially different effects. All right, so that was the study guide. Hopefully this helped. If you have any questions, feel free to email me and I'll be happy to answer them. I know this video kind of goes on, but feel free to fast forward or move backward whenever you need help with things. All right, I'll talk to you guys later. Bye. Okay, what are we going to do for UA time today?
Oh my God.